Hello students, welcome to my YouTube channel Biology with Miracle Teacher. In this class, we are going to learn T.S.of anther. In the previous class, we learned the anther has two lobes. This is one lobe, this is another lobe. Each lobe has two microsporangia. It means totally two lobes have four microsporangia. Singular form of microsporangia is microsporangium. Okay, so here now we will learn in detailed structure or internal structure of anther. In this wall, first we will learn wall of anther. Wall of anther is formed of four layers. Okay, so this is the outer layer, that is known as, this is outer layer, it is known as epidermis. Here it is next layer, second layer. So this is known as endothecium. Here this is middle layer. Next layer is tapetum. Then inside, what is this now? This is known as sporogenous tissue. Sporogenous tissue. Okay. In this cross section, at the center, you can see this structure. This is known as connective. Connective means that this is sterile tissue. Okay. Now we will see that the layers of anther in detail. So in this structure you see, this is epidermis, this layer. Outermost layer is known as epidermis. It is formed of single layer of flat cells. It gives protection to anther. Next layer is known as endothecium. This endothecium is also made up of single layer. Okay, so here these cells are endothecium. In the endothecium, the cells are arranged radially. All the cells are compactly arranged. Okay, so that is here. This is cells are radially arranged. In between the cells, in between these endothelial cells, they have some thickening substance that is known as callose. This callose is a thickening substance. Because of these thickening substances, this wall becomes thick. These walls become fibrous. So it becomes rigid. Okay. So third layer is known as, this is middle layer. Okay. This endothecium, functions of endothecium, if you see, that is dehiscence of anther. Dehiscence of anther. Next layer is known as middle layer. So all these three layers, the main functions of all these three layers is dehiscence of anther. So here in this endothecium, callose substance is present. No? This callose substance make this wall very thick. Means that makes the wall fiber. Okay, fibrous it becomes. This is the wall. Okay, so this wall, these three layers form this wall. So this wall is very thick, fibrous and rigid. Okay, so when the anther gets matured, it becomes soft. It becomes this thickness. Callose thickness will be dissolved by a substance, that is callase enzyme. Okay, so this thickening is everywhere, it is not having the same thickening. Okay, so at the place of dehiscence region. So this is dehiscent region. Okay, this region is dehiscence, line of dehiscence. If you see this structure, here you can see this is lines of dehiscence. Line of dehiscence. This line of dehiscence, so will be, that is when the anther gets matured, this dehiscence will gain this place in this region. That is cell wall will be burst and microspores will be released. Okay, so this wall will become delicate at this region where it is lines of dehiscence. Dehiscence region, this wall become thin. Okay, delicate. Then fourth layer is known as tapetum. Structure of tapetum, if you see, so this is pyramid shape. Tapetum structure, if you see, that is structure of tapetum. It is pyramid shape. Cells are pyramid shape. Have dense, that is dense cytoplasm. Many nucleus, so multinuclei. How it becomes multinuclei? This nucleus in the cell, nucleus will divide. Means a karyokinesis takes place, no cytokinesis takes place. Karyokinesis takes place, no cytokinesis takes place. Okay, next you see there is, it has many, they are polyploidy. They are polyploidy. And next structure you see, that is all the tapetal cells show increase in the DNA content. Okay, DNA content is increased. This is structure. Then functions of tapetum. Functions of tapetum means this layer is very important layer because it is nutritive layer. Nutritive layer. It nourishes this layer nourishes the pollen grains. Nourishes the pollen grains. Okay. So now you see here, this is sporogenous tissue. This sporogenous tissue means the cells of sporogenous tissues. The cells of sporogenous tissues undergo meiotic cell division to form microspores. Okay, when this anther becomes mature, the tapetal layer secretes an enzyme, that is callase enzyme. This callase enzyme dissolves, that is the callose substance. Okay, so this all of the anther become thin, they become soft. Okay, as well as the wall is delicate at dehiscence region. So this anther wall bursts and microspore releases. Okay, so the microspore releases as tetrad or 4 microspores together, arranged together and form tetrads. Microspore tetrads release. This tetrad is formed by callose substance. Okay students, thank you.